Hello dear students, welcome to the class. In this session, we are going to learn about the tissue systems which are present in the plant body, and also we are going to learn about the anatomy of root, anatomy of stem and anatomy of leaf. Okay, we'll be reading about the sections which are given in NCERT. So let's begin. If I ask you a simple question, what is the meaning of tissue system? What you are going to tell me? You already have learned about tissues, right? Group of cells which are having common origin and which are performing common functions, they are called as tissues. Together they are working as a unit to perform a particular function. Likewise, in a plant body, you have different types of tissues, right? So in order to understand them more easily, we can group them under one more category, which is called tissue system. What is the meaning of tissue system? Tissue system is nothing but it is the, you can say it is the tissues as per the function they are performing in the plant body, and based on where they are located, you can group different types of tissues under different tissue systems, right? And basically in the plant body, you only have three types of tissue systems. Okay, so what are these three types of tissue system? First is epidermal tissue system, second is ground tissue system, and third is vascular tissue system. You can see in NCERT lines they have mentioned that tissues vary depending on their location in the plant body. In different parts of the plant body, you will find different types of tissues. Okay, their structure and function also depend on the location, which kind of tissue you are going to see in the plant body and what function they are going to perform. This also depends on where they are located. Okay, now based on this, you have three types of tissue system: epidermal tissue system, ground tissue system or fundamental tissue system, and vascular or conducting tissue system. Now you try to assume that you have a small plant, right? You have a small plant like this. Okay, fine. And now you cut the cross section of this plant. Okay, take a transfer section of this plant. Okay, and try to see that based on location, how many different types of tissue systems you can see. So let's suppose I am cutting this stem, right? You can think about any structure. I am cutting a stem here. So when I will cut the stem, I will see the outermost layer. Okay, outermost layer which is protective in nature, which is covering the entire plant body. That will be considered as the first type, which is mentioned here, epidermal tissue system. Okay, epidermal tissue system. This epidermal tissue system is protective in nature. It is covering the entire plant body, right? And whatever features you can observe on this epidermis, that all will be considered as a part of this epidermal tissue system. For example, you are going to learn about stomata, epidermal cells, and you are going to learn about root hairs and trichomes, right? So these all you can see and observe on this outermost layer, which is there for performing protection, and some other functions are also there which you are going to learn. So this together is called as epidermal tissue system. It is the outermost layer, protective in nature. Then after that, if you will go in the center, right, in the center you might see that there are certain structures which are there for conducting water and food throughout the plant body. Okay, so these structures are called vascular bundle, and together these vascular bundles, they make vascular tissue system, or you can say conducting tissue system, right? This is the third type. Okay, so fundamentally you can see, or you can say universally, in all the plants you have epidermal tissue system outermost, and in the center you will see conducting strands which are made up of vascular bundles, xylem and phloem. Together they make vascular tissue system. And everything from here, okay, everything except epidermal tissue system, okay, I can say everything like this entire layer, okay, this entire area, okay, which is except the epidermal tissue system and except the vascular tissue system, it is called the ground tissue system, or you can also consider it as fundamental tissue system, because it is a filling tissue, right? It is the majority of the plant body is made up of this tissue system. So can you see the position and location where these different tissue systems are present? The outermost body of the plant is covered or surrounded by a protective tissue system, which is called epidermal tissue system. The central part of the plant body has some conducting strands, or you can say those tissues which are helping in the conduction of water and food. Okay, they together make vascular tissue system or conducting tissue system. And except this epidermal tissue system and conducting tissue system, rest everything is included under another type of tissue system which is called fundamental tissue system or ground tissue system. So you understood the position, their locations, right? Now let's learn about the very first type, that is epidermal tissue system. So if I talk about epidermal tissue system, you can see it is mentioned that it is the outermost covering of entire plant body. Entire plant body is covered by epidermis. Okay. Now this epidermal tissue system has different you can say structures here. So you will be studying first about epidermal cells. Actually epidermal cells or epidermis cells, they make this entire layer, epidermis. Okay, so epidermal cells, then on epidermis you will see some specialized cells which are there for gaseous exchange and transpiration. So you will be studying about that structure which is called stomata, right? And then there are certain extensions, certain appendages which you can observe on epidermis. They are called root hairs and stem hairs or trichomes. Okay, so you will be studying about these epidermal appendages. So these are the structures which you have to study. Let's study about epidermis first. NCERT writes about epidermis that this is the outermost layer of primary plant body. Can you explain me what is the meaning of this line? Why NCERT has emphasized here primary plant body? Please remember, whenever inside a seed embryo is present, you all know, whenever embryo develops, it first forms the primary plant body, right? Embryo develops into a young plant, right? That young plant is green in color and it is just a result of the primary meristematic activity. Primary permanent tissues have been formed there. So the primary plant body will be having cells which are covered, outermost layer of the plant body will be covered by epidermis. Okay, but when the plant is mature, not young, okay, primary plant body means they are talking about a young plant. Okay, when the plant is mature, okay, when the plant is mature, at that time secondary growth will happen and then the epidermal cells will be replaced by certain other type of cells, like you can say cork cells, which are dead cells, right? So you have to be very careful if in statement they are giving you a statement like outermost layer of the mature plant body is epidermis, that will be wrong in some cases, right? So it is present as the outermost layer of all primary plant organs, right? So it is present in the primary plant body. Then it is made up of elongated, compactly arranged cells which form a continuous layer. Just assume that epidermis is that layer which is meant for protection. Okay, you also have your skin is that layer which is meant for protection in your body, right? So assume that. Can you bear any kind of space in between the skin cells in your body? No, right? Because if it happens, what will happen? Many parasites, many pathogens can enter in your body. Many foreign cells can enter in your body and they can cause any kind of disease or discomfort to you, right? So as a barrier it is present, it is meant for protection, right? So it is protecting you from various kind of pathogens, right? Just like that, plant body is also protected from various kind of pathogenic attacks. So individual cells of epidermis cannot have any kind of intercellular spaces. What is the meaning of intercellular spaces? Space in between the cell. That is, space is absent, right? That's why it is mentioned it is a continuous layer without any intercellular space, right? So I'm writing here no intercellular spaces. Okay, there is no intercellular space. Okay, you have to remember this. Cells are compact, they are elongated, they are arranged continuously. Okay, now usually the epidermis of the plants they are single layered. What is the meaning? Usually means mostly. Okay, usually means mostly. Mostly the epidermis is single layered, but there are few cases where epidermis can be multi-layered also. For example, in Nerium, one plant is there where you can see multi-layered epidermis, right? But that is out of NCERT. So if they write, if they can give a statement question here, what they can write? They can write that epidermis is always single layered in plants. That will be wrong. Epidermis is usually single layered. It can be multi-layered also in few cases. Understood? Now the cells of this epidermis layer, they are living cells, they are parenchymatous. Remember, you have learned parenchyma is a type of simple tissue in the previous class, right? So it is parenchymatous. Parenchymatous cells are living cells, right? What is the content or composition of the cell wall of parenchyma? It is having cellulose, right? So the cell wall is thin, it is having cellulose, and it has a small amount of cytoplasm because there is a large vacuole in the center, right? So these are some characters of the epidermal cells which you have to remember, that epidermal cell, it is elongated cell, it is compactly arranged, right? All the cells are compactly arranged, they form a layer which is called epidermis. This epidermis usually is single layered structure, not multi-layered, but sometimes it can be multi-layer in few cases, right? Then each and every cell, if you will consider epidermal cell, it is a parenchymatous cell, it is a living cell, cell wall is composed of cellulose, and it is having peripheral cytoplasm because it is having a large vacuole in the center. Okay, small amount of cytoplasm it is having and large vacuole is present in the center. Now, epidermis is often covered by a waxy thick layer which is called cuticle, right? See, they are covered on top with the help of a waxy covering. Why? Because see, plants always have a stress associated with them, that is, if water is not available in the soil, what will happen? The plant will get dried and it will die, right? So to protect the plant from this desiccation, this dry condition is called desiccation. So to protect the plant from desiccation, all entire layer of the plant body which is covered by a waxy coating which is called cuticle. Maybe you have seen on some leaves it is present, right? It makes it very shiny. So that is the thing we have to remember. Now this thick waxy coating covering which is present above the epidermis is called cuticle. If a question is asked that cuticle is present in each and every part of the plant body, what you will think? What you will write? No. Cuticle is absent in the roots. Why? Because what is the work of root? Root has to absorb water, right? And if it has to absorb water, can we cover that epidermal cell of root with a waxy thick cuticle? No. Cuticle is hydrophobic in nature, right? Wax. If you take wax, if you take candle and you put water on that candle, can it, you can say, can it pass through that wax? No, right? So you have to understand this, that wax is something which is hydrophobic. Cuticle is hydrophobic. As a result, if there is any kind of cuticle layer present on the cells of the root epidermis, what will happen? Water will not enter in the root, right? So that's why cuticle is absent in the roots. Another thing you have to remember, in hydrophytic plants, okay, hydrophytic plants, hydrophytic plants, okay, in hydrophytic plants also cuticle is absent, okay, cuticle is absent. Why? Because these plants are already in the water, right? They don't have any kind of water loss problem, right? They don't have any kind of water stress. They are not having any problem of desiccation. They have enough water. They can absorb water by their roots all the time, right? So they also do not require any kind of cuticle. Most of the hydrophytic plants, they don't have cuticle. So roots don't have cuticle, hydrophytic plants do not have cuticle. Okay, rest aerial parts of the plant, they are covered by waxy covering called cuticle. Okay, now epidermis you understood, all the points from NCERT covered. Now is stomata. In this epidermal tissue system, or you can say when you will look at the outermost layer of the plant body, you will see certain pores are present there, right? Not everywhere in all the parts of the plant, but most of them, they are present in the leaves. Okay, so what are those pores? See, stomata is the name of the pore which you can see here. Okay, these pores actually are some specialized structures which are called stomata. So stomata are the structures which are present on the epidermis of leaves. And if I ask you a question that stomata is found only in the epidermis of leaves, no. Along with the epidermis of leaf, it is also present in the young dicot stem, or I'll write young stem. Okay, young stem. Okay, which has not done any kind of secondary growth. Okay, so on young stem also few stomata can be found. Few. Okay, here you will see more stomata. Okay, fine. Stomata are very important structures because they regulate gaseous exchange. Okay, you know that plant uh, do gaseous exchange, right? You know that carbon dioxide has to enter in the plant body and oxygen can get released from the plant body. So for this gaseous exchange, they need certain pores, and those pores are present here in the form of stomata. Also, plant has to lose a lot of water in the form of vapor, that is called transpiration, right? So that it can keep on absorbing the water from the ground. Okay, this depends on the pressure gradient, right? So water concentration is maintained in such a manner that from the leaves the water is always lost in the form of vapor. So the concentration of water, the particular water potential will be reduced, and as a result more and more water will be absorbed from the root. Okay, this you don't have this in your course, but it was there previously in one chapter which is deleted. So anyways, transpiration, at least the meaning you have learned in class 10th also, the loss of water in the form of vapor is called transpiration, and gaseous exchange, why it is needed, you know very well. Carbon dioxide is required by the plant for photosynthesis and oxygen is released as a product of photosynthesis, right? So that's why stomata are present. They allow these things, right? Now, if I am talking about stomata, okay, and there is one more term is stoma. Stomata, please remember, it is a plural term. Okay, if I'm talking about stomata, it is a plural term. Okay, and if I'm talking about stoma, okay, it is a singular term. Okay, stoma is a singular term. Okay, do not get confused. So each stoma, if I talk about one such structure, each stoma is made up of two bean shaped cells which are called guard cells. Okay, which encloses a stomatal pore, right? And in grasses, the guard cells are not bean shaped, they are dumbbell shaped. So I'll show you one figure along with this explanation. Just see this. This is what? This is a dicot stomata. Okay, stomata. And this is a monocot stomata. Even you can see it is given here also. And the same image was given in last year Neet, right? It was a PYQ for 2024. This diagram was given. You can check. Okay, so you can see very nicely here that they are talking about some specialized cells, which are these cells, right? These specialized cells are you can say bean shaped or kidney bean shaped, right? And you can see differential thickening in the cell walls here. The outer walls are thinner as compared to the inner walls. Okay, so this is what? This is the guard cell. Okay, you can see here they have mentioned this structure is guard cell. Okay, this is monocot stomata. Here you can see guard cell is labeled as this one. And you can see here thickening is in the middle part. This wall, this part of the cell wall is more thick as compared to the outer walls. Okay, fine. So when you have enough water here, I will label water potential like this. Okay, this means water potential, how much water is there in the cell. So if water potential is high, or simply you know this is called turgidity. Okay, if the cell is turgid, if the guard cell is turgid, what will happen? You will see lot of pressure will be given to outer walls, right? Outer walls are thin and elastic. Guard cells outer walls are thin and elastic. As a result, they will stretch, right? And if they will stretch, what will happen? A pore will be created here, right? Same way here, the turgid condition will result in the formation of pore here. So you can see that they have labeled stomatal pore here, over here. This is the stomatal pore. Okay, and I will label here, this is the stomatal pore which they have not labeled. Okay, so you understood? Guard cell is enclosing a stomatal pore. Now you can see plenty of cells which are present here also, right? Plenty of cells are present here. You can see these two cells are present here. Okay, these cells are connected with the help of plasmodesmata. Okay, here these cells are connected to these cells, you can see. Okay, here you can see this cell they have labeled as subsidiary cell. Actually it should be labeled as this, but fine, NCERT we have to consider that only. Okay, so this is the subsidiary cell here, and this is the subsidiary cell. Subsidiary cell is the cell which is surrounding the guard cell. Guard cell is enclosing what? Stomatal pore. What is the feature of guard cell? It is made up of cell wall which is cellulosic in nature, but the thickening is not linear or same throughout. The outer walls of the guard cells are thinner and the inner wall or the middle wall is thick. Okay. So outer walls of guard cells which are away from the pore, they are thin, and inner walls which are towards the pore, they are highly thickened. Okay. What is the shape of the guard cell of monocot monocot guard cell, and what is the shape of guard cell in dicot? Please write dicot stomata. Okay. Then guard cells, they possess chloroplast and regulate the opening and closing of stomata. Very important role. Okay. Guard cell, subsidiary cell, all of these cells are actually type of epidermal cells. Epidermal cells have got modified and they have developed these cells. You assume it like this. Okay. See, this is a leaf. Okay. Entire outermost layer of this leaf is covered by epidermal cell, right? So let's suppose this is epidermal cell, this is epidermal, every cell is epidermal cell. Some of the epidermal cells, they have got, okay, some of the epidermal cells, they have transformed themselves, okay, they have become like this. Understood what I am doing? Okay, some of the epidermal cells, they have transformed, okay, they have modified, okay, and they have become guard cells, they have become subsidiary cells. Okay, so what were these cells before? They were epidermal cells. These are the rest epidermal cells. I am writing EC. Okay, all epidermal cells, they are devoid of chlorophyll. No chlorophyll. Okay, even subsidiary cell also don't have any chlorophyll. But this guard cell is the only one which is green in color, which has chlorophyll, which has chloroplast, because this is cell organelle where chlorophyll is present, right? Here it is absent. Understood? So all the cells, all epidermal cells are devoid of doing photosynthesis except guard cell. Guard cell will do photosynthesis, right? Because it is having chloroplast. Very important line. Okay, and guard cell is the only cell which is regulating the opening and closing of stomata. If they ask a question, which among the following regulates the opening and closing of stomata, guard cell or subsidiary cell? It is guard cell. This can be a question. Which among the following cells will regulate the opening and closing of stomata? It should be guard cell. Okay, mechanism is, entire mechanism was there in one of the chapters, but now it is deleted, so I'm not going to talk about it. Now one more thing is mentioned: stomatal aperture or stomatal pore. Okay, stomatal aperture or stomatal pore, guard cell, subsidiary cell, together this all is called as stomatal apparatus. Okay, so subsidiary cell, guard cell and stomatal aperture or stomatal pore, together this all is called as stomatal apparatus. Okay, fine. Okay, now after stomata we have epidermal appendages to learn. What's the meaning of epidermal appendages? Any kind of appendage means hands, legs, all of these things are called appendages, right? So from epidermis, if you can see any kind of extension is coming out, that is called appendage, epidermal appendage. So you have two types of epidermal appendages. When you will see the roots of the plant, okay, on the root epidermis, you are going to see one appendage which you all know, unicellular appendage, which is called root hair. Okay, when you will see stem, on stem epidermis, you are going to see another kind of appendage which is called trichome. And another name of trichome is stem hair. Okay, so first please remember, root hairs are present on root epidermis and trichomes are present on stem epidermis, right? These are unicellular structures, but these are multicellular structures. Please remember. Okay, you can see the cells of epidermis bear number of hairs. The root hair are unicellular elongations of epidermal cells. Okay, for example, if I want to draw it, I can draw it like this. Okay, this is our, let's suppose, the cell of root epidermis. This cell itself will get extended like this, okay, and it will increase the absorptive surface area for absorbing more and more water, right? So this condition is there. It is an extension or elongation of the epidermal cell which help in absorbing the water and mineral from the soil. Now more absorptive surface area to absorb water and mineral, right? On the other hand, trichome is multicellular. What is the meaning here? If I simply draw it like this, so here as a result of cell division, you will find this structure getting formed. Okay, this is multicellular structure. Okay, you can see multiple cells here, right? So trichome is not extension or elongation of a cell of epidermis. No, it is formed from the cell division of epidermal cell. It's a multicellular structure. Okay, so stem hairs are called trichomes and they are usually multicellular. Okay, usually they are multicellular. Okay, only word if they are using, then this statement will be wrong. So please remember this statement, usually multicellular. They can be branched or they can be unbranched, right? They can be branched or unbranched. Like here I have drawn unbranched, but it can have branching also, right? It can be soft or it can be stiff. Very, very important, right? Sometimes the sometimes the stem hairs may appear very soft. Sometimes you will touch them, they will be very hard, stiff, right? They may even be secretory. So these are the functions. What is the function of root here? It is absorptive in nature. It is helping in absorption of water and minerals from the soil, right? What's the function of trichome? Trichome will secrete something. It is secretory in nature, right? Also, it helps in preventing the water loss due to transpiration, right? So when trichomes are present and stomata is deeply seated and above that you have these stem hairs or trichome, it will reduce. It is an you can say method by which plants they will reduce water loss. So please remember these functions. They will give you can say questions on these things. Okay, so please remember which one is the unicellular, which is multicellular, which is soft, stiff, which is soft and stiff, which can be branched or unbranched, which can be secretory, which is helping in the you can say preventing the transpiration. Right now, ground tissue system. What's the meaning of ground tissue system? I just told you ground tissue system is everything except epidermal tissue system and vascular tissue system. It is considered as ground tissue system. So all the tissues except epidermis and vascular bundles, they constitute ground tissue system. If you leave vascular tissue system, if you leave epidermal tissue system, everything else is ground tissue system. I'll write about it. See, outermost layer in the plant body, you will see epidermis. Okay, epidermis. After epidermis, you are going to see something which is called cortex. Okay, cortex. Fine, this is the next layer. After cortex, you will see another layer which is called endodermis. Okay, after endodermis, you will see another layer which is called pericycle. And after pericycle, you will see vascular tissues. Okay, vascular tissues. Okay, and then after that you will see in the center you will see pith. Now if you leave this epidermis and if you leave this vascular tissue, everything from here to here, this will be a part of ground tissue system. Okay, cortex, outermost cortex, epidermis, pericycle and innermost pith, right? Okay, pith is present in the center of the section. Okay, now sometimes cortex further can be differentiated. It can be differentiated as hypodermis, okay, cortical layer and endodermis. 3 layers of cortex are there, but this all will be discussed in different sections. So you have to just learn that these are the layers which are a part of ground tissue system. Okay, now see, sometimes you will see some layers, like most of the layers of cortex. Okay, so you will see cortex, most of the layers will be parenchymatous. Okay, pericycle can be sclerenchymatous, sometimes it can be parenchymatous, right? You will see that pith always parenchymatous, medullary rays always parenchymatous. So what you have to understand here, this ground tissue system is a type of tissue system which is made up of parenchyma also, collenchyma also, sclerenchyma also, or you can say this is made up of simple tissue system. I will tell you in different sections that hypodermis is parenchymatous or sclerenchymatous. So ground tissue system is made up of all different types of simple tissue systems. Understood? Okay, fine. Okay, now Parenchymatous cells usually are present in cortex. Okay, this is not fixed. They have written the term usually here. Usually, most of the time, cortex will be parenchymatous, pericycle will be parenchymatous, pith and medullary rays will be parenchymatous. I have not written medullary ray there. I will show you in sections. In the primary stem and in the root. In leaves, the ground tissue system is different. They don't have these layers, cortex, endodermis, pericycle. These all layers are present in stem as well as in root, right? In stem and root, you have these things. But if I talk about leaves, okay, in leaves, the ground tissue system is having another name which is called mesophyll tissue. Okay, when you remove epidermis, when you remove vascular tissue, everything in between the upper and lower epidermis will be comprising of a tissue which is called mesophyll tissue system. It is a type of ground tissue system in leaves. Because it is having chloroplast, it will be doing photosynthesis. Understood what's the meaning of ground tissue system? If they ask you a question, which among the following tissue system is made up of all types of simple tissue system? What will be the answer? Ground tissue system. Okay, fine. Now vascular tissue system. What's the meaning of vascular tissue system? Vascular tissue system, it is made up of xylem and phloem. It is conducting tissue system, so definitely it will be having xylem for transport of water, phloem for transport of food, right? So xylem and phloem together, they will form vascular bundles, and these vascular bundles, they will form vascular tissue system. Now very important part of this topic is arrangement of vascular bundles. Okay, NCERT gives you these three diagrams. In these three diagrams, you have to learn the fixed arrangement of vascular bundle in different sections. In the first figure, what you can see, as per NCERT, this is mentioned that this is the radial arrangement of vascular bundle. Okay, what is the meaning of radial arrangement? Can you see that the red color bundle which you are going to observe, it is xylem labeled here, and the blue one is phloem, right? Now try to notice that the vascular bundles, okay, they are present on two different radii, right? They are present on different radii. Present on different radii. What's the meaning? Xylem is present on this radius and phloem is present on outer radius, right? They are not on same radius. Okay. Next thing you can notice, okay, that they are alternating to each other. Alternating to each other. You can see here xylem, then you can see phloem. Xylem, phloem. Xylem, phloem. Okay. Understood? And each bundle is separate, or you can say individual xylem and phloem bundles. Xylem and phloem are not present together as one single bundle. They are independent, right? You can see a bundle of xylem, then another bundle of phloem, then another xylem. They are not together. Xylem and phloem is not present as single individual bundle, right? They are separately present. This condition is called radial arrangement of vascular bundles. Okay, I hope you understood. Okay, this arrangement is called radial arrangement of vascular bundle. Now, in which particular section you are going to see this? In which plant section you are going to observe, or which plant section is having this radial bundle? So please remember, radial bundle is observed in all roots. Okay, all roots. Whether it is dicot root, whether it is monocot root, you will see only radial arrangement of vascular bundle. Right? Now, here these two types which are given here, they are called conjoint. Okay? These two are called as conjoint. Okay, what is the meaning of conjoint? Conjoint vascular bundle mean, can you see here phloem and xylem are present together in one bundle? Yes. So what I will write here? Phloem and xylem present as single bundle. Okay, present together as single bundle. Okay, phloem and xylem present together as single bundle. Okay, they are not separately present. Okay, now this one is called conjoint closed. Okay, this one is called conjoint closed, and this one is called conjoint open. Okay, this kind of arrangement where phloem is always towards the outer side or facing the periphery, and xylem is facing the inner side or towards the pith side, this is called collateral also. So I can write this is conjoint and this is collateral. Okay, what is the meaning of collateral? Collateral means phloem on outer side. Okay, xylem on inner side. So these are conjoint, collateral. This is conjoint closed, this is open. What is the meaning of closed? Closed vascular bundle means here cambium is absent. Cambium is absent. What's the meaning of cambium? Cambium is another name of meristem. Okay, it's a primary meristem which is present inside, but function is like a secondary meristem. They also help in dividing the tissues and making secondary permanent tissue. That's why I'm saying function is like secondary. Here the cambium is present. Dividing cells are present. So if cambium is absent here, can this do secondary growth? No. So I'm writing no secondary growth. Right? But here cambium is present, dividing cells are present. Cells will divide on both the sides. They will add new cells on both the sides. So what I will write? Here I will write secondary growth occurs. Okay. What is the example for conjoint closed? For conjoint closed, we have an example. Please remember all monocot stem and all leaves. Whether it is dicot leaf or monocot leaf, these two you have to remember, monocot stem and all the leaves. And here all the roots are having radial. And what you will remember here, dicot stem is having conjoined open type of vascular bundles. I hope this is understood to you. Okay, so please see this very carefully, because when we are going to do sectional studies, these things are very important here. Okay, you can observe conjoint closed is present in monocot stem and all the leaves. Conjoint open is present in dicot stem. Right? What is the meaning of open? Cambium is present. What's the meaning of cambium? It is doing secondary growth. What is the meaning of secondary growth? Secondary growth means increasing the width of the plant or making wood. Do any kind of leaf increase in the width or it will make any wood? No. So what kind of vascular bundle it will possess? Conjoint closed. Okay. Grasses, monocot stem, all of these structures, will they have any kind of secondary growth? No. So which kind of vascular bundle they will have? They will have conjoint closed. Understood? All the roots, whether they are monocot roots or dicot roots, they all will have radial arrangement of vascular bundle. Understood? Fine. And I have not talked about cambium here, because cambium is not present in between the vascular bundle, because there is no such arrangement of conjoint condition here, right? So cambium will get formed in the dicot root, but there is another method of that we will explain later. Understood? Fine. Now let's talk about the anatomy of dicot and monocot root. You can observe two sections on the screen, right? They are very important sections. You have to learn about the differences which are present in them as well as the similarities which are present in them. If they want to make any kind of difficult question, they can give analytical question based on these differences and similarities. Okay. Now try to observe. They have talked about that they have given the section of sunflower root in dicot, right? So on the left side you can see this root which is given to you, it is dicot root. Okay, and on the right side you can see this is the monocot root. Fine. You can see the outermost layer here is epidermis in both, right? You can see here it is mentioned as epidermis, here also it is mentioned as epidermis. Can you tell me what is another name of root epidermis? It is also called epiblema. Okay, another name of root epidermis is epiblema. Okay, here this is also called epiblema. Fine. So root epidermis is called epiblema. You cannot use this term for stem epidermis. Fine. Epidermal cells are okay, this is single layered, or I will write it separately. Okay. Common things I am writing here. Okay. So here I am writing common characters. Okay. first I will be writing about epidermis or epiplemma. Okay, if I talk about this layer, this is single layer. Okay, no intercellular spaces. No intercellular spaces. Root hairs are present. Okay. For absorption. Water and minerals, right? Because root hairs are extensions of epidermal cells, so they will increase the absorptive surface area, you can see. But here they are more numerous in dicot as per the diagram, and lesser as compared to dicot in monocot. But that is not that much important, right? So you have understood about epidermis. Next layer to epidermis is cortex. Okay, next layer to epidermis is cortex. Okay, can you see any kind of you can say differentiation in the layers of cortex, that different layers are marked here? No, you can see after epidermis here they have mentioned cortex here, right? And all the cells are looking alike, right? So cortex, you have to remember, this is multi-layered, not much differentiated. Okay, this is not differentiated. Only endodermis is present as the last layer of cortex. Okay, it is not differentiated. Okay, then parenchymatous in nature. Okay, epidermis is also parenchymatous. I am not writing because you have learned epidermal cells are parenchymatous cells. So cortex cells are also parenchymatous. Okay, here intercellular spaces are present. Intercellular spaces, small intercellular spaces are present, right? And there are also parenchymatous thin walled cellulose containing cell wall. So you can see small intercellular spaces you can observe here, right? So this you have to remember. Now the last layer of cortex is called endodermis. Okay, what is endodermis? Endodermis is last layer of cortex. This is also parenchymatous. This is always remember the endodermis is barrel shaped. Okay, endodermal cells are barrel shaped cells. Okay, no intercellular spaces. No intercellular spaces. There are no intercellular spaces at all. Okay, after that you have to remember here the cell wall is having deposition of suberin. Okay, very important. Cell wall is having deposition of suberin. Suberin is hydrophobic in nature. Okay, water impermeable material. Okay, impermeable to water. Impermeable to water. Okay, fine. Okay, this thickening is present. Okay, this particular thickening which I have written here, it is present in the form of what? It is present in the form of strips. Okay, so I'm writing over here. Suberin deposits both on radial as well as tangential walls in the form of strips. And because the name of the scientist was Casper, that's why these strips are called Casparian strips. So Casparian strips are very important feature which is observed in which cells? endodermal cells. Okay, fine. So endodermis also understood. Okay, it is single layered. Single layered I have written? No. Okay. Single layered, no intercellular spaces, barrel shaped cells, parenchymatous, but cell wall is having, and this covering. This common to this is common in both the sections, right? Now after this, which is the next layer? Next layer is pericycle. There are differences in pericycle, but right now I'm writing all the common points. So if I talk about pericycle, okay, pericycle if I write here, please remember pericycle is few layered. It is not one or two layer, it is few layered. Okay, and it is parenchymatous, thick walled cells. Okay, cells are living cells, parenchymatous cells, but thick walled cells. Please remember this. Okay, fine. And now if I talk about the dicot root, in the dicot root, please remember the pericycle has two functions. Okay, the cells of pericycle are responsible for two things. First is pericycle is responsible for forming, or it develops lateral roots. Okay, the branches of the root. Okay, like this is, let's suppose, primary root, secondary root will form from here, then tertiary root. So all this is happening from which layer? Pericycle layer, right? So it is responsible for development of lateral roots. Okay, another another function of pericycle is it forms vascular cambium for secondary growth. Okay, it forms vascular cambium for secondary growth. After that, we have monocot root. Monocot root, only one function, only one, it develops lateral root. It does not help in any way in developing vascular cambium because cambium is not present in the monocot root. They don't do secondary growth, right? That is the difference. So I hope you understood up to pericycle, right? After this, you have pith. Okay, one more layer is there which is called pith. Pith also has characteristic difference. Okay, pith you have to remember it is the last layer, or you can say parenchymatous. Okay, please remember in the dicot root, pith is reduced, or another term used is inconspicuous, right? But in the monocot, it is well developed, large pith, well developed, large. Okay, diagram is not clearly depicting that, but okay, fine, in the text it is mentioned that pith is large here, pith is very much reduced here. This area is called pith, this central part is called pith. Okay, now after pith you can see that there is some tissue present over here. So that tissue is called conjunctive tissue. Conjunctive tissue. Okay, what is conjunctive tissue? It is present between xylem and phloem. Okay, it is parenchymatous. Understood? These are the different layers which you can see here. Okay, conjunctive tissue you will see. I'll show you here. See this area. Okay, this entire thing is xylem, right? This red color structure which you are able to see, this is xylem. Okay, and these small cells, can you see here the small cells present here, small cells here, and smaller cells here. These small cells are called phloem. Okay, they make this phloem. Same condition in that diagram also. Okay, these areas are phloem. Fine. Okay, now this area, okay, this part is called pith, you can see, and here the cells which are present in between this xylem and phloem, okay, the cells which are present in between this phloem and xylem, these cells are called conjunctive cells. Okay, so here this tissue is called conjunctive tissue. Okay, same goes for this also. Understood? Fine. Now, can you find any characteristic difference? Because most of the things are same in both the sections. The majority of the differences you are going to observe are in the vascular bundles. Okay. If I talk about the vascular bundles, okay, vascular tissue. If I talk about this, in the dicot root, you will be having only 2 to 4 bundles. Okay. But in monocot, you will be having more than 6 bundles, and this more than 6 bundle is called polyarch condition. Okay, so what are the characteristic differences in the dicot and monocot stem? Okay, the characteristic differences are everything is more in the monocot root. Okay, you have larger pith, right, and you have polyarch vascular bundle. This is the difference. Okay, but as compared to this, in that case, what do you have? You have lesser number of vascular bundle and you have smaller pith. But there's one more difference here. Here pericycle is responsible for doing two things, making vascular cambium as well as responsible for originating lateral branches. But only lateral branches are observed by like lateral root formation is observed from pericycle. There is never vascular cambium formation by pericycle in the monocot root. Another thing I would like to tell you is the arrangement of xylem and phloem here. Okay, please remember xylem in the root is of exarch type. Okay, what's the meaning of this? Okay, xylem here in the root section, it is always exarch. Okay, what is the meaning of exarch? See, this is the arrangement of primary xylem. Okay, primary xylem means first formed xylem is of two types. I will write here first. Primary xylem is of two types. The first formed xylem is called protoxylem. Okay, protoxylem, and the next formed xylem is called metaxylem. After this, secondary xylem is formed. So protoxylem, when it is always towards the periphery, when the protoxylem is towards the periphery, okay, and the metaxylem is towards the center, this arrangement of our primary xylem is called exarch arrangement. When you will see the lumen of protoxylem, it will be very small, but the lumen of metaxylem will be bigger. So you will see from the pith, it is like this. This is protoxylem, this is metaxylem, and this entire arrangement is called exarch arrangement, which you are going to observe in these two sections. Can you see? The bigger xylem is towards the center, the smallest primary xylem is towards the outer side. This arrangement of the primary xylem is called exarch arrangement. All the roots, whether it is dicot root or monocot root, okay, dicot or monocot, they have this kind of arrangement. I hope this is understood and clear to you. These are the things which you have to remember in root section. Now let's learn about stem section. In this stem section, what you can see, this is the dicot stem. Okay, this is dicot stem, and after this dicot stem, you have this section which is of monocot stem. Monocot stem. Please see in the dicot stem, what are the differences which you can observe here? The first and the most important difference, you can see the arrangement of vascular bundles. They are arranged in a ring-like condition here, right? But the vascular bundles are scattered throughout, right? So this you have to remember. Okay. If I talk about epidermis, there is no much difference. Epidermis is single layered, usually single layered. Here also it is covered by cuticle when it is young stem. Here also it is covered by cuticle when it is young stem. Then after that, you can see here these layers are what? These are together except pericycle from parenchyma, endodermis. Together these are what? These are cortex, right? So cortex here is well differentiated. Cortex here in this section is well differentiated into three layers. Okay, three layers. The outermost layer is called hypodermis, and hypodermis is collenchymatous. Okay, inner to it, it is called cortical layer. Few layers are there, cortical layers. This layer is actually parenchymatous. Intercellular spaces are also present here, few intercellular spaces. And the last layer is endodermis, right? And endodermis is parenchymatous, but this endodermis is very special because it is having deposition of starch grains, right? And as a result, this endodermis is also called starch sheath. Okay, it is also called starch sheath. So cortex here is well differentiated into hypodermis, cortical layer, endodermis. You can see here epidermis, hypodermis, and endodermis. Epidermis is called you can see here hypodermis, endodermis, and this parenchymatous layer, which is cortical layers, right? Fine. Along with what type of simple tissue is making that also you have to remember. Okay, after that you have pericycle. Okay, pericycle is very important because pericycle here is sclerenchymatous, dead. Okay, and it is present as semilunar patches. It is present as semilunar patches. Okay, above endodermis and below phloem. What's the meaning? Let's suppose if I want to draw one vascular bundle here, so I'll draw phloem here and xylem here and cambium layer here, right? And on top of this, I will draw this pericycle, and above this pericycle will be endodermal cell or not, right? So this is that sclerenchymatous semilunar patch of pericycle, right? So can I say pericycle is continuous in the dicot stem? No, it is present as patches. Okay, as patches. Okay, is it living or dead? It is dead. Can it ever make any kind of a branch? No, right? Understood. After that, you can see phloem, cambium, and xylem. Okay, this is xylem, and if you see the xylem, protoxylem is facing the center or pith. You can see in the center you have pith also. Protoxylem is facing the pith, and metaxylem is present on the you can say outer side. Okay, this kind of arrangement, when the protoxylem is facing the pith and metaxylem is facing the periphery. This kind of arrangement of xylem, who will tell me, this is called endark arrangement. This is called endark arrangement. All kind of stem have this arrangement. Okay. Now, can you see here in the center you have a large pith? Yes, pith is large here, right? And there is a tissue in between the vascular bundle. You can see in a ring-like fashion, the vascular bundles are arranged, right? Ring-like arrangement of vascular bundles. Very characteristic feature of dicot stem, right? In between this vascular bundle, there is some tissue which is present here. This tissue is parenchymatous, and this is called medullary ray. You can also even call it as primary medullary ray. This is parenchymatous. Okay, you can see it is mentioned here. Okay, so these are the layers in dicot stem. Just try to see and compare this with monocot stem. Monocot stem looks so simple, right? Monocot stem has outermost layer epidermis. Okay, it will have hypodermis, but it is sclerenchymatous. Means dead. Okay, after that you have this ground tissue, which is parenchymatous. Okay, always remember, in the monocot stem, endodermis, pericycle, pith. All these three things are absent. You don't have endodermis, you don't have pericycle, you don't have pith, right? And just look at the vascular bundles. Vascular bundles, first of all, they are surrounded by sclerenchymatous, Okay, they are surrounded by sclerenchymatous bundle sheath cells. They are surrounded by sclerenchymatous bundle sheath cells and they are scattered throughout. They are scattered throughout the section. They are scattered throughout the section. So they are everywhere, they are scattered. And just look at their size also. There is a difference in size, right? Size is larger bundles are present where? Just see the image and tell me. Larger bundles are present in the center, right? And smaller bundles are present where? Towards the periphery. Okay, fine. Understood? Now one more thing is mentioned in NCERT, that phloem parenchyma. Okay, when you will see the vascular bundle, phloem parenchyma is absent in the monocot stem. Okay, and you have water containing cavities in the monocot stem. Water containing cavities, they are present. These are the things you have to learn to solve questions. Okay, fine. Just compare. Here the cortex is well differentiated. Here the cortex is not differentiated. It is having only this sclerenchymatous hypodermis. There is no endodermis. There is no nothing. Only one ground tissue is there, which is starting from here and ending up to here. Everything is filled by this ground tissue, which is parenchymatous. Okay. Each and every vascular bundle is surrounded by dead cell containing layer, which is called bundle sheath cell. Okay. So hypodermis is also dead and bundle sheath cells are also dead. Okay, then in between only ground tissue is there, which is parenchymatous, where intercellular spaces are also present. Endodermis, pericycle, pith, phloem parenchyma absent. Water containing cavities are present. Okay. Size of the vascular bundles are not same throughout. Central bundles are larger as compared to the peripheral. As you are moving towards the periphery, the size is getting reduced, right? This is what you have to remember. And do you have any cambium here? No, cambium is absent. The bundles are conjoint closed. And can you see here also, this is the larger one. Metaxylem is facing the outer side, and protoxylem, this is small one, is protoxylem, this is facing the center. So here the arrangement of vascular bundle, just like you have learned in the previous case, in this system it is endark, right? Protoxylem towards the center. I have written pith because pith is present here, but pith is not present here, so I'm saying center, right? The metaxylem facing periphery and protoxylem facing center. This is called endark. All stems have endark arrangement of vascular bundle. Okay. Now anatomy of dicot and monocot leaf. Okay. Now see, if I talk about dicot and monocot leaf, just tell me, a dicot leaf always grows like this. Laterally it is fixed or arranged, right? But a monocot leaf grows like this, okay, straight, right? So when this happens, what will happen here? This side will receive more sunlight, but in monocot, what will happen? All the sides will receive equal amount of sunlight or not? Understood. So what will happen? This side, okay, will be considered as dorsal side. And the hidden side, the back side, is considered as ventral side. Okay, this dorsal and ventral side is present. That's why this dicot leaf is also called as dorsiventral leaf. Okay, here this is a monocot leaf. Okay, in monocot leaf, what is the condition? Each and every side receiving same amount of sunlight. That's why no characteristic site is formed. It is called isobilateral leaf. Okay, that's why this dorsal side is always darker as compared to ventral side, which is lighter. And here all sides have same color. Understood? Okay, fine. Fine. Now if you have understood this, just see the section of dicot leaf. This is dicot leaf. In dicot leaf, you can see, if you take your hand also is covered by epidermis, right? This will be outer epidermis. This will be epidermis which is present on the dorsal side. This is the epidermis present on the ventral side, right? Just like that, leaf also is covered by epidermis on both the side, right? So this is upper epidermis is called adaxial epidermis, okay, and the lower epidermis is called abaxial epidermis, okay. You can see over here, okay. Now there's no intercellular space in between the epidermis, you can observe very nicely here, and there's a cuticle also present here. Can you see thick waxy coating here on both sides, right? Which side is receiving more sunlight? This side is receiving more sunlight, above side, right? So if this is receiving more sunlight, definitely this side will have more chance to do photosynthesis, right? That's why you can see the color of this side is dark. So mesophyll is here differentiated into palisade mesophyll and spongy mesophyll. Okay, you have mesophyll cell, you have mesophyll tissue which is differentiated. Okay, differentiated mesophyll tissue. Okay, mesophyll tissue is differentiated into, okay, palisade mesophyll and spongy mesophyll. Can you see palisade mesophylls? They are elongated, compactly arranged, more chloroplast, right? More green in color. And spongy mesophyll, can you see? They are having some air cavities also. They don't have that much chlorophyll as well, right? So because of the position, one side is receiving more sunlight. That's why the arrangement is like this, and that's why the characters of the mesophyll differ, right? So that is the reason that mesophyll is differentiated here, because the leaf, dicot leaf, is not receiving equal amount of sunlight from all the sides, right? So you have palisade mesophyll and spongy mesophyll. Now, along with that, you can see vascular bundles covered by bundle sheath cells, xylem and phloem you can see, right? Another thing, you can see the arrangement of stomata. You have negligible amount of stomata on upper epidermis, but you have plenty of amount of stomata on lower epidermis. Why? This is a method by which the plants can reduce the chance of transpiration, because this plant is receiving more sunlight from this side, so they have reduced the number of stomata on this side. Okay, anyways they have to do gaseous exchange, but they don't want to do transpiration, right? So more like they don't want to do more transpiration, so more number of stomata is present on lower side, so that normally gaseous exchange is possible and some amount of water also can be lost, not too much. Okay, fine. Just see the labelings of these diagrams. That is very important, because they are sometimes giving you some figures from here and they are just writing it. What is this ABC? Just to tell which among the following is correct match for this label A, right? Let's suppose substomatal cavity. They will not ask you what's the function, but they will definitely give you as a label. So please revise these diagrams very thoroughly, right? Now just look at this. This is what? This is a monocot leaf. Okay, in this monocot leaf, you can see there is no differentiation of this mesophyll tissue. This is not differentiated. What is the reason? The reason is that from all the sides, you are receiving equal amount of sunlight. So there is no need of any differentiation. And you can see the distribution of stomata is also almost similar, right? You have one stomata labeled here, one is given here. So you have similar distribution of stomata on both the upper and lower epidermis. You have same type of mesophyll throughout the leaf. You don't have any differentiation in the mesophyll, right, in the monocot leaf. But very important thing, there are some cells, okay, there are some adaxial epidermal cells, okay, they have become modified. They have become modified for what? They have become modified to serve very important function, right? How they will get modified? They will become colorless, they will become empty, and they will become large, right? They will become colorless, they will become empty and large, and they will form a cell which is called bulliform cells, also called motor cells. Okay, when these bulliform cells, they will be empty, they will be large, and they will be colorless, they will be free to have a lot of water. So when these bulliform cells are turgid, okay, when these bulliform cells, okay, they are turgid, what will happen? They will have lot of water and they will keep the leaf exposed. Okay, But when the same bulliform cell, it will become flaccid, okay, or they will lose a lot of water. What they will do? They will allow curling of leaf, or you can say rolling of leaf towards inside. Okay, you can assume a paper, okay, and try to roll that paper from both the sides. What will happen? The exposed surface area will be now hidden. Okay, so by this what will happen? If the leaf is exposed, then stomata will be exposed or not, okay, to do transpiration. And when the leaf is curled, what will happen? The stomata will become hidden, right? And as a result, the plant will not lose lot of water, right? So this is the method, this is an adaptation by which monocot plants, they can adjust or they can live in such high temperatures also. And you can say when there is lot of desiccation condition, when there is very less water, when water scarcity is there, then also the leaves, what they will do, grasses and all, okay, mostly this is found in grasses, okay, monocots like grasses. Okay, what they will do, they will be having certain cells which are called bulliform cells, which are modified upper epidermal cells or adaxial epidermal cells. They will lose the water and they will allow the curling of leaf or rolling of leaf towards the inner side. The surface area of the lamina will be reduced, and as a result, the number of stomata will become hidden. Okay, all the stomata will become hidden, and then transpiration will be reduced. Okay, it will reduce transpiration. This thing will reduce transpiration. Okay, fine. Now let's learn how much you have understood, okay, by solving certain questions. See, parenchymatous structure with intercellular spaces is, okay, see, epidermis is also parenchymatous, endodermis is also parenchymatous, cortex also parenchymatous, pericycle also in some cases parenchymatous. But which is having intercellular spaces also? This can never have intercellular space. This can never have intercellular space. This also does not have. Then cortex is the answer or not? Cortex is having intercellular spaces or not? Remember about root. Root, I have shown you that figure. Lot of intercellular spaces were there in the cortex. Select the correct feature with respect to trichomes in shoot system. Okay. If I talk about trichome, I'm talking about what? I'm talking about stem here. Right? Stem here. Okay. They are usually unicelled? No. They can be branch or unbranched? Yes. They may be secretory? Yes. Soft or stiff? Yes. They helps against transpiration? Yes. So what is the answer here? All except A. Okay. All except A. Pericycle of root is never sclerenchymatous because it, what's the meaning of sclerenchymatous? Sclerenchymatous means dead. Okay, and pericycle of root, why it can never be dead? You know the reason, right? They have to divide and they have to form lateral branches, or maybe in dicot root they even have to do they have to make vascular cambium. So if they are already dead, how they can divide, right? So what is the reason? They does not act as mechanical tissue because it is the place of origin of root branches. Okay, it give rise to root hairs. No, it give rise to root hair. No. So second option is the answer, right? Which is not true for monocot stem. What is not true for monocot stem? Monocot stem has hypodermis, which is sclerenchymatous or not. Correct. They have presence of water canals in pith. Okay, let's come back to this option. Conjoint collateral closed vascular bundle, correct. They have a presence of bundle sheath, this is correct. What is not true? This is not true. Do they have any kind of water canal and do they have any kind of pith also? They don't have any pith, right? So this is wrong. Okay, which of the following vascular bundles are always open? Always open. Okay, conjoint, radial, collateral, bicollateral. It should be bicollateral. Okay, this is out of NCERT, but I will tell you. Let's suppose this is a vascular bundle, and in a bicollateral type of vascular bundle, which is there in some of the plants like cucurbita and all, you'll be having two cambiums, right? And you'll be having two phloems, and xylem will be here. Okay, so collateral, what I told you, phloem is always on outer side, then you have xylem. And in this vascular bundle, if you have cambium, then it's okay. Sometimes you cannot have cambium, then it will be considered as closed vascular bundle. Remember? Okay, this is what? This is conjoint open and this is conjoint closed. Okay, This is collateral vascular bundle also because phloem is on the outer side. This is also collateral because phloem is towards the outer side. But this is conjoint, bicollateral because on both the side you have a phloem, and this is always open, always open. Okay, they have cambium always. Okay, this kind of vascular bundles are rare and you are going to see this in the family Cucurbitaceae. Okay, watermelon family, pumpkin family, there you can see this. Okay, but this is out of NCERT. See, radial cannot be the option because see, you can actually eliminate the options here. Conjoint can show you open and closed both condition or not, so you can remove this. Collateral can show you both conditions or not. Radial in monocot route you cannot have vascular bundle open, but in dicot you can have open to secondary growth, right? So you can eliminate and you can get the answer. Anyways, no problem. This is an extra information which you can remember. Bicollateral vascular bundles means xylem is sandwiched in between the phloems. Outer side also phloem is there, inner side also phloem is there. Okay, in between you have xylem and they have two cambium always, right? So this is there, and in which family this exists? In watermelon family, Cucurbitaceae family, this happens, right? But this is not given in NEET syllabus. Fine. Monocot stem differs from dicot stem in having what? Okay, so monocot stem and dicot stem both have endark xylem, correct? Both have well developed pith. Monocot stem does not have pith at all, right? Polymorphic vascular bundle, yes. What is the meaning of this term polymorphic vascular bundle? Polymorphic means vascular bundles are not looking same. The outer ones are actually smaller as compared to inner ones, right? Remember, vascular bundles are scattered and outermost are size wise they are smaller, right? Peripheral one are smaller. So this is polymorphic, multiple forms, right? Collateral vascular bundle, that is same in both, right? Now, origin of roots, origin of lateral roots and formation of cambium is related to. Okay, so you will give me the answer. I have repeated this many times. In the root, there is one layer which is responsible for making lateral roots as well as cambium, that is called pericycle, right? Now, transfer section of this is a PYQ. This is a PYQ. Transfer section of plant part showed polyarch, radial, and exarch xylem. Okay, polyarch condition, radial. Radial, so you understood. This is talking about root. Polyarch, you understood. Poly means multiple, like more than 6. So it is talking about monocot root, right? And exarch xylem also you understood. It is talking about root. So this is an example of monocot root. Okay, grass leaves curl inwards during the dry weather. Okay, select the appropriate reason from the following: closure of stomata, flaccidity of bulliform cell, shrinkage of air space in the mesophyll, or stylosis in the vessel. Okay, it is the flaccidity of bulliform cells because grasses, they have some cells which are colorless, empty, large. When they will lose all the water, they will allow this curling inward. Because of that, what will happen? The exposed surface area will get reduced and the stomata will become hidden and transpiration will be checked. Okay. Casparian strips. Casparian strips are there in which of the following sections? Like in which of the following layers? It is there in the endodermis. Okay, so these were our 10 questions which we needed to discuss. I hope you have understood. Okay, please keep NCERT along with this if you are watching this, and try to watch it on 2x or 1.5x speed so that you can revise it very fastly. Along with that, I have not added or included secondary growth in this. If you want secondary growth, I can make another you can say video for that, as NTA has removed that topic from current syllabus, so I have not included it here. This chapter is completed as per NTA syllabus, but I will also teach you secondary growth in a one short session. Okay, thank you everyone. Share this video please and help others. Thank you. Bye.